I thought you said that if we showed up on one button, we have to show it like on one calculation.
For this, so you show like this. You only have to show once, right? You're showing all the calculations that you do for the lab, and then here. But if you're doing additional problems, you've got to show them all. Okay. Show them all.
Okay, thank you.
Show them all. Good morning, lab report. Do you want it?
Yeah. I'm scared.
Why does this one not have a break? Oh, because it goes with that.
Oh, that's what. You know what? Mine was literally right in front of me. I guess I don't have to be nervous.
Cover your nose.
Oh, sorry.
I know.
I just got so excited.
Sucks.
It does.
It does. That's why I got contacts. It really is.
I've been waiting over a month for my contacts. The company has not shipped them yet. It's like,
what do you do? I know. I know. I know. All right. What's that? Oh, thank you. I looked at the Moodle link and I was like, there's a reason why I have to go to Moodle. Why? And I didn't go. So thank you for saying that. Because I always do that at the very beginning. Okay. Reading. 10/18/21. Whoa, that didn't work. Okay, happy Monday, everybody. Please go ahead and sign in on the spreadsheet that I almost forgot to put up. Oh, and I did forget my thing. Okay, well, guess it's mass day. I'll have to speak extra loud. Okay, so what kind of a car do sheep drive? A Lamborghini. Stole my joke. A Lamborghini. I thought it was cute. I thought that was cute. Okay. So the last thing we were talking about is our 5 major functional classes of proteins, right? We just barely got into chapter 6. So now we're going to kind of dive right in. Full screen. All right. So the first set that we're really going to discuss are metabolic enzymes. Now, metabolic enzymes can be proteins, and what else can they be? What else can be enzymes? RNA. So either proteins or RNA. And these are going to catalyze and control our metabolic flux, right? So what does that mean, that they're going to control metabolic flux? So basically what catalysts do are to provide that optimal environment for the really rapid conversion of reactants to products, right? That's the whole name of the game, to make it really fast. So they're going to catalyze our biochemical reactions. And a lot of times we're talking about energy conversion. So anybody look ahead and know some of the energy conversion pathways that we're going to discuss, like glycolysis, the citrate cycle, right? All that kind of stuff. So what do they do for us within those pathways? They're going to lower the activation energy. Now, how they lower the activation energy, we're going to talk all about in chapter 7. We are going to explain that in excruciating detail, not excruciating, but in lots of really interesting detail, right? Really interesting. I think it's cool. To end up increasing our rate of protein or product formation. It's not necessarily protein, but rate of product formation. So this is really, really important because if we didn't have enzymes, we would all be dead. The rate at which we have to complete enzymatic reactions is enormous. So enzymes are going to increase the rate of that reaction by 100,000-fold. Oh, I cannot talk and write at the same time. It's bad, bad. So that's a lot. And remember that we're not going to alter our equilibrium concentrations because are we really operating at equilibrium in a cell? No, we're not operating at equilibrium. A lot of times we calculate delta G at equilibrium, but then what's different in the cell? If we want to shift a reaction to go in one direction, what do? Deplete the products, right? If you want to shift it in the forward direction, deplete the products. If you want to shift it in the reverse direction, what do? Deplete the reactants. And you can change that shift or change that delta G value just because of your concentration of reactants and products. And remember that through all this, our enzymes are unchanged, right? They're regenerated so that we can use them over and over and over again. So we'll talk about how we use enzymes for synthesis, and we will also talk about how we use enzymes for degradation. So, and the other thing to remember with these metabolic enzymes, right? Usually it's not just one step in a metabolic pathway. That's why we call it a pathway, because there are tons of steps. And so what you're going to see is a lot of times that these metabolic enzymes are only one part of a really big complex, right? And the reason that we want that really big complex and having one enzyme do one thing and then another enzyme in the complex do another, and then another enzyme in the complex do another, is that that first enzyme that goes from reactants to products, that product is a reactant for the next enzyme. So the next enzyme uses up the product, and what does that shift our reaction? Forward. Shifts it forward to the products. So that because it's sequential, we can drive all of those reactions in that one metabolic pathway. That's really important. Really, really important. So they're usually part of a metabolic complex to drive pathway forward. Okay. All right. So let's look at one example. Now, I have to say that for metabolic enzymes, they usually do a pretty good job of naming things. Y'all know that gets on my nerves when they name things poorly. So basically, when they name metabolic enzymes, they name it using the name of one of the substrates and then what it does. So if we have malate dehydrogenase, what do you think it does? Takes away hydrogen from what? From malate, right? Oh my gosh, they named it well. So that's exactly what happens. And so what you can see here, you'll see different diagrams in the book, but basically a lot of times what they do is just to highlight the portion of malate that's important, right? So we're highlighting right here, this is the hydrogen that we're going to use. And in the presence of nicotinamide, nicotinamide adenine dinucleotide, that's the NAD+, we're going to remove that hydrogen. Where does the hydrogen go? We're going to make what? NADH. So we're oxidizing malate, right, into oxaloacetate. And then in the reverse, what's happening to NAD? Being reduced. So a lot of these metabolic reactions are a combination of oxidation and reduction. One substrate is being oxidized, the other substrate's being reduced. And most of these reactions are reversible. So that you can go backwards and forwards, right? So in the next chapter, what we're going to do is we're going to look at these enzymes. Now, the malate dehydrogenase is the enzyme here in, what color is that? I don't even know, cyanish, teal. Oh, teal's a good one, yeah. So that's the teal color is the enzyme itself. And then inside that pocket, you have the coenzyme NAD+. So this NAD+, if it's not present, this reaction is not going to proceed. And so we call it a coenzyme. It's required for the enzyme function. And so we'll look specifically at the enzyme active site and see how some of these enzymes catalyze these chemical reactions, which is really, really insanely important. So we'll devote a whole chapter to that. All right, the next set of proteins are the most abundant proteins in living organisms. They are structural proteins. So you got to think about structural proteins kind of twofold, right? One is we want to maintain the integrity inside the cell. So when you look inside the cell, you'll see things like actin and tubulin, those kinds of cytoskeletal proteins. And they're going to maintain a shape and also to promote changes in shape so that maybe it's a white blood cell that needs to migrate to a site of infection, right? So you'll have the cytoskeleton changing the cell shape so it can move. Also, you have cytoskeletal proteins that are secreted, that are found in the extracellular matrix, right? That give a framework for individual cells to live. Have y'all looked at bone tissue in anatomy? Anybody? Some people? You've seen like right? So most of the bones, right, are not actually living tissue. The living tissue is excreted by the bone cells, but you don't have, if you look at mass of your bones, right, most of it is excreted tissue and not really cellular. Y'all understand what I'm saying? Yeah, okay. So sometimes you excrete it and then it provides a framework for cells to live in or for organs to be supported in, right? So they're kind of twofold. All right, so let's talk about a subset of those. Let's talk about cytoskeletal proteins. Cytoskeletal proteins are inside the cell, give you cell shape, cell migration, cell signaling. The biggest ones that are inside the cell here, these are inside the cell, are actin and tubulin. So these two are the most abundant proteins inside of animal cells, the most abundant. And they're both able to self-polymerize, which means that if you put monomers of actin or tubulin together, they will spontaneously turn into filaments, which is insanely cool. Collagen, on the other hand, is one of those proteins that is excreted into the extracellular matrix, right? So this is they are all structural proteins. Two of them are found inside, one is mainly excreted. So if we look at my favorite protein on the entire planet, right, this is actin. Now, I actually studied actin in non-muscle cells, but in all of your muscle cells, actin and myosin are the two most abundant proteins because they form the contractile unit that allows us to contract all of our muscles. And we'll talk about this in the very last section, how actin and myosin work together in order to allow you to move locomotion wise anywhere you want to go, which is really, really pretty interesting. Okay, tubulin. Tubulin is another cytoskeletal protein that's really important. But the difference with actin and tubulin, actin is more of a skeleton in terms of how we think a skeleton works, right? What does the skeleton in your body do? It gives you shape and structure, right? Allows your muscles to have something to push and pull against. So the same is true about actin. It gives you that 3D shape of your cell. But you got to think about inside the cell, some things are made in one portion of the cell and have to be transported somewhere else. Think about a vesicle, like a neurotransmitter. So let's say you have a nerve cell and you've made this vesicle and it's full of neurotransmitters. And you got that electrical signal that says, go ahead and release your neurotransmitters at this plasma membrane at the synapse, right? How do you get this vesicle to the plasma membrane? You hook it onto a myosin motor, and it's really cool. The myosin motor like grabs onto it like it's Santa Claus. That's what I think of. It holds on to the vesicle and it walks along a tubulin structure, and then it dumps it out. Do you want to see? Yeah. Okay, so if you Google, it's NIH, the living cell, I believe it is, the living cell animation. It should come up. Inside the living cell. Let's see. This is like one of the coolest videos I've ever, I absolutely love it. Okay. So this is an example of a white blood cell that's inside of a blood vessel. And this is kind of how the white blood cell will stop moving in the blood vessel and actually translocate and enter the tissue. It slows its rolling down because it's having those connections with cells on the surface of the blood vessel, and it'll actually enter the tissue. So now they kind of look at different structures within the cell, and it goes on and on and on. But what I want you to see, what I really want you to see is here. Okay, so we're going to go into the cell, and what all these are actin filaments, and the actin filaments are cross-linked to one another. And that cross-linking is like scaffolding, and it helps with structure, right? You have lots and lots of support within the actin filaments. And so what you'll be able to see is that it will self-assemble and disassemble on its own. This is it assembling, right? Those are the actin monomers coming together to make a double helix actin filament. Isn't that awesome? It's like so insanely cool. And you have actin binding proteins that associate with the actin to either sever it, so this is a severing protein, or to stabilize it. It can do either. And so you see them disassembling in the distance, right? So now this is tubulin. Tubulin makes microtubules. And so you see they self-assemble and disassemble in a similar way, except this makes a tube instead of a coiled helix. And so these are the roadways inside the cell. You can transport things from one part of the cell to another using these microtubules. So here is, I believe, it's either dynin or myosin, where it catches the vesicle and then it walks it. Oh, that is exactly how it works. Isn't this so cool? Now this is happening in your cells all the time. Yeah, that like flip down the wall. Yeah, Okay, so if you feel interested, you should watch the whole video with the narration and everything. It's super insanely cool, but that was what I really wanted you to see out of it, just for what we were talking about. Okay, so now do you understand how they're molecular roads?
That, I guess the actin and the microtubule, they are one of the parts are able to come together. What exactly makes them so special that
they can self-assemble? So it's the thermodynamic, it's the kinetics of their interaction. Once you you'll have things bump together, right? Inside the cell. You got to think, and that's the one thing I don't like about that video, is that things looked really far apart. Did you notice that? That's crap. It's for visual representation, because otherwise, if you were looking at what's really in the cell, it would be really hard to see everything. So you have to think that these actin monomers There are so many of them that they very easily bump into one another because there are so many. So once they kind of bump into each other in the right orientation, once you get 3 together, you start forming a filament. Just because it's more stable to be in that polymerized state, right? So it's all about thermodynamics and kinetics and that kind of thing. It's really cool. Yeah. If you're really interested in it, I could we could have a very long conversation about it. All right. So microtubules are really important for roads in terms of transporting things from one part of the cell to another, but they're also really important in pulling those chromosomes apart, right, in cell division, which you don't see in that video. So the one thing I do want to say before we move on about models is that every model has flaws. Every single model that you will ever look at is not right for everything. But some models are useful, right? So it's not perfect what we saw, but it's a really good representation, really good. Okay. All right, collagen we said was really important. And primarily it's a big component of all of our connective tissue. So we talked about bones, right? Bones and teeth. Collagen is excreted, and the way that it's excreted, it makes basically like tubes, right? And so all of those stacked one on side of the other give really a lot of strength to your bones, right? Same thing in tendons, in cartilage, all of the parts of our skeleton that actually contain lots and lots of strength that comes from collagen. Collagen is a very, very strong protein. Okay, so this is type 2, right? That was type 1 was cytoskeletal. Now our second type are transport proteins. So transport proteins are all about facilitating movement, right? That's what transport says. So you're going to find a ton of them in the plasma membrane. So think about a transport protein. If it's going to be in the membrane, it's going to span that entire membrane, right? From lipid bilayer to lipid bilayer. So it's going to permit things to cross the membrane that would not normally cross on their own. So what things can cross the membrane without any help? Small particles that are non-polar. For example, oxygen. Do we need an oxygen transporter? No, it just diffuses across the membrane, but not everything does. So polar substances, charged molecules, all of those things need help to enter and exit the cell. We're going to look at glucose a ton in the future. Glucose is a ring sugar. And what does it have on that ring? That's like organic, right? Lots of hydroxyls, right? Which are very polar. So do you think glucose is going to get in and out all by itself? No, it's not. Okay. So we're going to have to transport it in and out. So there are two basic classes of transporters. We have passive transporters and we have active transporters. So passive transporters take advantage of what process? Diffusion. Diffusion, right? So this is another way to say diffusion is you have a chemical gradient. And when you have diffusion, you go down the gradient, right? Down the gradient means you go from areas of high concentration to areas of low concentration. Then you can have active transport. What's the difference between active transport? Requires energy. And we'll talk about energy. It's not always ATP. Sometimes there's a different form. So it can either be ATP or you can take advantage of an ionic gradient. And I'll show you how those two work. But really, in a passive transport, do you think we have to change shape in order for it to happen? No. How about active? Absolutely. Active is always going to have some kind of conformational change. Conformational change, right? That's going to open or close this channel. To open, close the channel. So that's going to be really, really important. You're going to see how much structural changes are super insanely important. So let's talk about passive first. We said energy independent, right? Does not require ATP or anything. And so all it's doing is allowing our molecules to move in response to that difference in gradient. So you got high concentration on one side of the membrane, low concentration on the other. They move to areas of low concentration. So there are two basic categories of passive transporters. We have porins. Porins are small molecules for small molecules like water. You heard of an aquaporin, right? That's for water. Glycerol is another example, but water is kind of the biggest one. And then we have ion channels. So what do you think ion channels are going to transport? Ions like protons, like potassium ion, like chlorine ion, like calcium ion. Now, calcium's really, really, really important. You know how we contract all of our muscles? It's all about calcium. But calcium for an ion channel will only go from high concentration to low concentration. But in order to get your muscles to contract, you actually have to pump a ton of calcium into the interior of the cell. And it's not going to happen by diffusion only. So we can't really do that to make muscles contract, like our heart, like our skeletal muscles, with passive transporter. We need something like an active transporter. So this is a calcium ATPase. Does it say it? This is a calcium ATPase. I don't know why it's doing that. ATP ASE. So a calcium ATPase. And so this is going to allow you to pump from areas of low calcium concentration to areas of high concentration. Do you see how this is against the chemical gradient? So you're going to have to do for muscle contraction, you're going to have to pump. It's like a concentration that's like 10,000 times higher, right? You're never going to make that happen spontaneously. So you're going to have to use an active transporter to transport that. And then you're going to have to be able to transport the calcium back. So think about your heart. How many times does your heart beat? How often does your heart beat? Yeah, so more than once a second, right? So in under a second, you're going to have to pump all these calcium ions in to the basically cytosol from a vesicle inside the cell and then back again. Y'all, that has to happen all the time. Because if your heart stops beating, what happens? That's it, you're done, right? So it's happening all the time. So you're using a ton of ATP to do this. So in this case, for a calcium ATPase, can you guess, is it going to be dependent upon ATP hydrolysis or an ionic gradient? ATP hydrolysis. Hey, we're naming some things well, right? Okay. So the next set, so this is what, number 3? All right. So cell signaling proteins, right? So this is all about what we talked about in the 2 chapters ago, where we have an extracellular signal and we need to turn that into an intracellular signal. So it's going to start with some kind of a receptor on the outside of the cell. And there are a couple of different receptors that can do this. You can have G protein coupled receptors. You can have receptor tyrosine kinases, and you can also have growth hormone. We're going to add that one. Hormone receptors. We're going to talk in detail about G protein coupled receptors. So you'll hear me call them GPCRs. That's a G protein coupled receptor. And then receptor tyrosine kinases. Those are going to be our 2 really big ones. But you also have things that are nuclear receptors. What's a nuclear receptor? Wait, what was that? Well, so we're in the nucleus. Not really receptors on the nucleus, because you got to think, if you have a nuclear receptor, you're going to have to pick up a signal that can pass through the plasma membrane. If it can pass through the plasma membrane, what else can it pass through? The nuclear envelope, the nuclear membrane, however you want to say that. So where do you want to have nuclear receptors? I don't know, in the nucleus. You want them in the nucleus. These are things like transcription factors. TF, transcription factors, right? You want to be able to turn gene expression off and on from extracellular signals. So transcription factors can bind to these ligands that go all the way through the plasma membrane, all the way through your nuclear envelope, and you can turn expression off and on. I mean, that's really important. All right, so the big ones, we have membrane receptors, we have nuclear receptors. Now, you also have intracellular signaling proteins. Intracellular signaling proteins are things that are basically downstream from our receptor, right? So our receptor gets activated, and then you need intracellular signaling proteins in order to propagate that signal. So how did we say typically inside the cell we turn things off and on? What modification do we do? Phosphorylation. Woo. So this is phosphorylation, dephosphorylation. So what do we call those enzymes that do that? Kinases and phosphorylases. Kinases and phosphorylases, right? Kinases phosphorylate, phosphorylases dephosphorylate. Okay. You can also have, so intracellular signaling proteins can be kinase phosphorylases. They can also be adapter proteins. So sometimes they'll bind to the activated receptor. And just because the activated receptor has changed conformation, now it can or cannot associate with an adapter protein. So these are, we'll call them intramolecular, CLECU, molecular adapter proteins. Right? So when the receptor becomes activated, it changes its conformation inside the cell. Now it can associate with the protein, adapter protein, or it stops associating with the adapter protein. Either way will create a downstream signaling effect. But the adapter protein changes the signal from being just stuck to the membrane to being able to signal further down in the cell. So it's that transition. And then you can also have site-specific proteases. So site-specific proteases, right? Because you want to degrade things at certain times dependent upon a particular signal. And that will change your cell's expression pattern and all kinds of things, right? So those are all different types of intracellular signaling proteins. So let's go and talk about the major membrane receptors. The major membrane receptors are G protein coupled receptors. So G protein coupled receptors are a lot of times those that bind to epinephrine related ligands. When do you produce epinephrine? Fight or flight, right? Epinephrine, y'all have heard of that before, right? So if you get really scared, what do you start making? You start making epinephrine, and it allows you to run faster, gets you more oxygen because your breathing rate increases. It dilates your pupils so that you can see a little bit better, right? It does all kinds of things that will help you to survive a fearful situation, right? There are tyrosine receptor tyrosine kinases. These, the example we're going to talk about is an insulin receptor. And an insulin receptor is very important. Why? What does insulin tell you to do? It tells you that when your blood sugar is high, to go ahead and pull that glucose into the cell to be used. So diabetics right, don't get that insulin signal. Either they don't make the insulin themselves or their cells are not receptive to the insulin that they do make, right? Either one is same problem. You don't take in glucose, and so you end up with really high blood sugar, and you could die from that. And the last type of the growth hormone receptors, these form dimers inside of the membrane. So like you have two halves to the receptor, and when the receptors bind to the ligand, they dimerize. And when they dimerize, that induces a downstream signaling event, right? So each of these three behave a different way in the membrane, and we'll talk about each of them in kind. So here's an example of the very last one, the growth hormone receptors that dimerize. So erythropoietin is a hormone. Anybody know what it's for?
Red blood cells.
To make red blood cells. Have you heard about, what's his name? Lance Armstrong, right? And the big blood doping thing that swept through the cycling, or what do you call that?
The Tour de France.
Yeah, the Tour de France. So basically with blood doping, there's two different ways to blood dope. Well, actually there's three. You can give yourself this hormone, erythropoietin, and this will stimulate red blood cell production, right? So for example, if you went and gave blood, your body would then make its own erythropoietin to bring your red blood cell count back up to normal. But it takes a while. It takes a month, you know, a little more than a month, maybe about 3 months, in order to get fully back to your original levels. So you could take the hormone, right? So a lot of times, instead of taking the natural hormone, they'll take a synthetic hormone. So what happens if you're an athlete and you have extra red blood cells? You can go for longer, right? Because you have a higher oxygen carrying capacity. So that's one way. Sometimes they also give their own blood and then after 3 months receive it back. So that then increases their red blood cells so that you have higher oxygen carrying capacity. It can be really dangerous though. Anybody know why? Nope, you're giving your own blood and you're getting your own blood. You're good. You will change the viscosity of your blood. Yeah, so if your blood viscosity gets too high because you have too high of a red blood cell count, red blood cells can't get through the smaller capillaries and you end up having basically blood clots and you die. So, you know, it's not fair and you could die, so don't do it. All right. So this is just an example where you have these two different parts of the receptor, and once the hormone binds, right, it forms the complex so that you have the complex actually meeting on the intracellular side. And once you have the conformational changes in the intracellular side, then you can propagate that signal throughout the entire cell, right? Okay, that was a side trip. All right, so nuclear receptors. Nuclear receptors, like we said before, are transcription factors. And we want transcription factors to be able to respond very quickly to external signals. So the most common types of ligands that will interact with nuclear receptors are steroid-based ligands, right? So hormones are derivatives of steroid, right? And so the most common illustration of that is the sex hormones, estrogen, progesterone, right? All of those cells, not all of the cells, but all of those hormones will actually interact with the transcription factors to change how the cells express their protein profiles, and then the cells will have a different effect. And you can see this in the women's hormonal cycle, right? So that's what controls it. It's a steroid hormone that goes straight into, past the plasma membrane, past the nuclear envelope, and goes and associates right with those transcription factors. Okay, so you can have intracellular signaling proteins. So intracellular signaling proteins, remember, these are the ones that we said, kinases, phosphorolases, right? So look at our examples, adenylate cyclase, protein kinases, All of these are ACEs, right? So what do you think they're going to use? ATP in order to accomplish their conformational changes and things like that, right? So these are all of our molecular switches, and molecular switches have to undergo a conformational change. That's how it always works. So sometimes you're going to have a receptor being activated, and that receptor itself will have kinase activity. And sometimes it won't be until you get to a downstream adapter protein where that adapter protein has kinase activity. But you can have kinase activity at any point in the pathway. So protein kinases, remember how we said the amino acid side chains are able to be phosphorylated or dephosphorylated, right? And which ones did we say could be phosphorylated and dephosphorylated? Serine and threonine are the two most common, right? What do they have on them that allows them to be phosphorylated? That oxygen, right? The OH is an OH group. And when the H comes off, the oxygen can bind to a phosphate group, right? So these are the enzymes that will alter serine and threonine amino acid residues on our protein targets, right? So you have enzymes that will phosphorylate your protein targets, but they will only do it once that receptor is activated. If the receptor is not activated, then your enzyme isn't activated and nothing happens. So it's all dependent upon that initial receptor binding activity. So in these pathways, we'll say like the receptor becomes activated. It associates with this adapter protein, which then eventually activates these kinases. So the downstream kinases in this pathway, that's what these could be. The MAP kinase proteins, so myogen activated proteins, protein kinase A, PKA, SARC kinases, phosphoinositide 3 kinase. So these are all downstream kinases that are going to work on substrates that are inside, or targets that are inside the cell after receptors activated. It's a lot of new terms in this chapter. Okay, the last one, right? What are we on, 4? 5. Genomic caretaker proteins. If you don't maintain the integrity of your genome, what do you have? Mutations, right? And the degree to which you have the mutations is going to tell you if your cell is going to survive or not. So you have a lot of different genomic caretaker proteins that are going to maintain your genome from one cell to the next. Has to be perfect, right? When you're replicating your cell, but you also have to be able to access certain parts of the genome to transcribe genes, to replicate the DNA, whatever it is. So there are proteins that are involved in replication. Which ones are important in replication? Polymerase, primase. Ligase does what? Seals a nick. Seal a nick in DNA, right? Topoisomerase, definitely, right? We talked about that before. Topoisomerase is one, we're talking about DNA replication. Is one that what? Unwinds the DNA, right? And relieves those supercoils on the DNA. Repair and recombination. DNA ligase is going to be very important in DNA repair and in recombination, right? We use recombination events to fix problems in DNA. We said that's how CRISPR works, right? That we have that template there. CRISPR will go in and cut the bad portion out, and then we'll use that template to make a copy and to fix the genome, right? So you have some that are important in repair, some that are important in replication, and you have some that are really important in gene expression, right? How many different polymerases do you have in the cell? Well, you got like 10 different DNA polymerases. You have lots of different DNA polymerases. And then you also have RNA polymerase, right? So you have a different enzyme that's going to make new DNA than the enzyme that's going to make new mRNA, right? But they're both polymerases, but they do different jobs. So here's an example of one genomic caretaker protein. So this genomic caretaker protein is called the REC-BCD complex. So if you look at this complex, this was isolated in bacteria. If you look at this complex, what this complex will do is as it slides along the DNA, it actually opens up the replication fork, right? So do you see how we now have a piece of single-stranded DNA here? So what this will go and do is it will look for errors. And once we know that there's an error, what it can do is it can actually go in and cut it and then use the template to make a new matching strand for that portion of it. So it's there to really scan along the DNA and open it so that the repair mechanisms can go in and fix it. So a lot of times you have tons and tons going on all at the same time. And so all of this is ATP dependent, right? Okay. So those are our big categories, right? Our big categories of all the different proteins inside the cell. Now with the rest of the chapter, we're going to go in and we're going to look at some specific examples of those different classes. All right. So the very first one we're going to talk about are myoglobin and hemoglobin. Myoglobin and hemoglobin are transport proteins. So have you heard of myoglobin and hemoglobin before? Yes? Okay, so what's the difference between myoglobin and hemoglobin? Myoglobin is in our muscles. Hemoglobin is in our red blood cells. So go in and put red. Right? They're both there in order to transport oxygen. It's all about oxygen transport. Myoglobin is for muscles. Hemoglobin is for red blood cells, right? So all of them are going to bind to molecular oxygen, O2, right? The one we're going to talk about in the most detail is hemoglobin. And so think about where hemoglobin is going to go in the pathway, right? When it's in the lungs, this is where it's going to pick up oxygen. Then it's going to transport that oxygen to wherever in the body it needs to go. It's going to drop it off where oxygen levels are low. And then it's going to be transported back to the lungs in order to pick up the next one, right? So it's a cycle. And so both proteins bind reversibly to iron in a porphyrin ring. And I'm going to show you what this looks like. Because we need something that's going to bind to the oxygen. So we have iron here that's going to associate with our oxygen. So if you look here, this is the structure of the porphyrin complex inside of heme, inside of hemoglobin, right? So this is a porphyrin ring. What do you see in the porphyrin ring? For what? For nitrogens. So no amino acid side chains can bind to oxygen. So basically we had to figure out another way to bind to oxygen. So if we could bind iron within the porphyrin ring, then the iron can bind to the oxygen. So the oxygen will only bind to the prosthetic group with the iron in the reduced state. So tell me about reduced and oxidized states of iron. So if we're iron 2+, what's our blood like? What does it look like? It's really, really red, right? So this is bright red. But what happens when we have iron, when we have cells that die? Like, you know, you get your blood drawn. What happens to the brightness of the blood after a while? It gets dark. It almost goes like that dark, like brick red, like that darker red. So what do you end up with? Fe3+, right? This is the brownish, I'll call it brownish red. And it's because the iron's being oxidized. And that's what gives it that brown color. But in order for that to happen, right, you're no longer carrying oxygen because we're in the reduced state, right? So these are dead cells. Okay. So if we compare the structures of hemoglobin and myoglobin, they're very different. They have a very similar function in different places, but they do look very different. Myoglobin is a single polypeptide chain that has one heme group, only one. So there's your porphyrin ring, right? And with your iron sitting right in the middle. The nitrogens are typically those blue, the blue dots. So that's how you can always find it. You see the ring and you see the blue dots as in the nitrogen atoms, and then you have the iron in the middle. Hemoglobin is that same general structure of a polypeptide chain, but it's actually 4 polypeptides. And the 4 polypeptides have 2 alpha and 2 beta subunits. So if you look here, right, the alpha and the alpha, the beta and the beta. So myoglobin looks like either 1 beta subunit or 1 alpha subunit. They're really, really similar. So there are 4 heme groups here. So see how we have 1 heme? We have 1, 2, 3, 4. So these are 4 heme groups, right? So this is a really, if we have 2 beta subunits and 2 alpha subunits, what will we call this myoglobin protein? It's really a dimer of heterodimers, right? Because if you have 2 alpha subunits and you have 2 beta subunits, we have a dimer of heterodimers. So if you look at these, even though they have that same heme group, they have the same porphyrin rings, there's really only about 20% amino acid identity from one to the other. That's a really, really low percentage, right? Do you remember we said in the beginning of the year that you have to have really high percentages in order for it to be homologous, for the proteins to be homologous, but every now and then there are exceptions to the rule. This is one of those exceptions, right? Even though we have only 20% identity, they function really, really similarly. Okay, this is a really good animation. You should go in and you should watch the animations. Do y'all watch the animations? Yes? Please go and watch the hemoglobin animation. It's really, really, really helpful. Okay, so when we talk about how hemoglobin works, we're gonna really zoom in on the enzyme itself, and we're gonna look at how it binds to the oxygen. So if you look within the molecule, do you see this silver part? This is part of a ribbon structure of the enzyme, right? And we're saying, okay, on residue 7, we have what amino acid? Histidine, right? So we have this histidine. Remember, histidine is my ring structure. I have my nitrogens, and one of my nitrogens is bound to a hydrogen, right? So what can it participate in? Hydrogen bonding. Well, O2 can participate in hydrogen bonding. So we have one histidine that functions on one side of the oxygen, and then we have another histidine that functions on the other side of the oxygen. So if we look at histidine 8, now the E and the F have to do with what part of the molecule they come from. So remember how we said there were 4 polypeptide chains? They come from different polypeptide chains. But we're looking at them because both of these interact with the oxygen. So we call one of them the distal histidine, and we call the other one the proximal histidine. The proximal histidine is associated with the heme group. The distal histidine is not. So we have another histidine, but this histidine is going to interact with the iron. Y'all see that? Now, the iron is going to make a covalent bond with the oxygen. And what that actually ends up doing, if you're going to make a covalent bond, what are you sharing here? Electrons. So if the electrons are shared between the iron and the oxygen, this iron actually takes up less room. And so you have a different structure within the whole hemoglobin, whether oxygen is bound or not. because if oxygen is unbound, this iron atom actually takes up more room and it causes a puckering. And that just that small change will actually change the whole 3 dimensions of the entire hemoglobin molecule. So watch. Okay, so this is our without oxygen. The heme, the heme, which is really here, right? It was this ring structure. Ooh. Right? And this is inside of the ring. And why do I keep wanting to go that way? Go this way. Right? We have this ring and inside of the ring is the iron, right? So they're just not drawing the whole ring. So if the ring is not bound to the oxygen, it actually binds to the nitrogen. And so what actually ends up happening is that that iron atom takes up more space and it actually ends up protruding out a little bit. And so that actually pushes on this histidine and changes the conformation of the F helix. The F helix basically gets shifted this way. Now, if you have oxygen present, the oxygen is sharing electrons with the iron. The iron's taking up a little bit less space. And so that heme group, that prosthetic group can be completely linear, completely flat. So then what happens to the F helix? It basically gets pulled inward. If you are pushing and pulling on a helix within a whole molecule, right? You have all these side chains that are interacting within the 3-dimensional folded protein. If you start moving a whole helix inward and outward, what's going to happen to the whole molecule? You're going to have propagated conformational changes, right? So either way that you look at this, right? You have deoxyhemoglobin or you have oxyhemoglobin. Globin. You're going to have a certain conformation of the entire overall protein in deoxyhemoglobin, and you'll have a different conformation in oxyhemoglobin, right? You have one small movement of one helix. You're going to cause propagated conformational changes. See what now? In the entire protein. Right? That's how this works. You have one small change that induces a propagated conformational change in the entire molecule. And then deoxyhemoglobin functions one way and oxyhemoglobin functions another way, which is what we're going to talk about next time. All from sharing a few lonely old electrons.
So the grade and homework committee said that she was taken out. It didn't address.
No, I know. I'm trying to talk to the publisher about, I don't want to take it out. I want to just give everybody some credit for it.
Oh.
So I'm trying to talk to the publisher about doing that, and I don't think they quite understand what I want. So I might just end up having to like take it out. So, but I really want to just give you credit for it. Like if it's worth X number of points, you worked on it. I want to give you
Right. I'd answer for the variable.
Right. So either I'll be able to go in eventually, give everybody credit on that one, or I'll have to pull it out. So give me a few more days to work with them. See if they can.
Also, is your test going to be similar format?
Yeah, it's similar. It's going to be some multiple choice, it's going to be some fill in the blank. It's going to be, there aren't so many problems, so it'll be more discussion. Like the discussion problem section is going to be similar.
You might have like 3 or 4.
So it'll be a few more discussion and less problem.
Okay, will you give us more as well? Like, you know how you gave us about 6 and you choose 2?
Oh, to choose from?
Well, you do like, would there be still 6, but you just have to choose 4?
Yeah, you'll still be able to choose. You'll still be able to choose. I like that. I like giving that to y'all.
Okay.
I think then like, you know, it's like, oh my God, I only studied this one that she asked about, right? So it gives you that little bit of cushion on which ones you study and which ones I ask for. This is gonna be different every semester, you know. So I try to do that for you.
All right, thank you.
Yeah. Let me see. Anybody need anything on Zoom? I think everybody, nope, people are still there. Haley and Tammy, y'all need anything? Good? All right, awesome. Let me shut this down because the next professor's coming through. So I can take questions to the hall if we need to.
Maybe I was thinking